Hello everyone and welcome back to Dr. Wasif Academy. In previous lecture we have discussed about photosynthesis mechanism, the research history of photosynthesis and leaf structure. We have also discussed about chloroplast and chlorophyll, which are main players of photosynthetic process. I'm sure that you have understood the what is difference between chloroplast and chlorophyll, and if you are asked to differentiate between these two terms, that is chloroplast and chlorophyll, you can easily respond to that question. In continuation of previous lecture, today we are diving into limiting factors of photosynthesis, which is a very important topic of biology and especially in relation to photosynthesis. Under this topic, we shall discuss that how various environmental factors including light, temperature and carbon dioxide affect photosynthetic mechanism. The topic about essential minerals will be discussed later, okay, in tomorrow lecture. So my dear students, stick around until the very end to improve your ability and knowledge for getting higher grades in your exam. So let's get started. Imagine you are baking 100 cakes, but you only have one oven. No matter how much flour or sugar you buy, you can't bake faster because the oven is your limiting factor. So, in biology, a limiting factor is any environmental variable which is in short supply and directly restricts the speed of photosynthesis. If you increase this factor, the rate goes up. If you do not, the rate stays flat or plateaus, even if you increase everything else. The first major factor, which is a limiting factor, that is light intensity. As you know that light provides the raw energy needed for the very first stage of the reaction. At low light levels, like at dawn or in a shaded area, light is the limiting factor. As the sun comes out and light intensity increases, the rate of photosynthesis climbs. But notice the graph here. Notice this graph. Eventually, it levels off. Okay, so at this point, giving the plant more light would not help. Why? Because the ovens are full. The plant is now limited by something else like the amount of carbon dioxide or the temperature. So here is the direct link you need for your exam. Light energy is used in the chloroplast to split water molecules into hydrogen atoms and oxygen gas. So if light intensity drops, this water splitting stops immediately. The plant runs out of the hydrogen and ATP, which it needs to build glucose, and the entire process will be shut down to a halt. So one of the most common experiments you will see in your biology, it involves measuring how light affects the gases a plant breathes. Now remember, plants are living things, so they carry out respiration 24 by 7. However, they only perform photosynthesis when there is light. To see which process is winning, we use a special liquid called hydrogen carbonate indicator. This indicator is incredibly sensitive to carbon dioxide because when carbon dioxide dissolves in water, it becomes acidic. So by looking at the color of the liquid, you can see different colors of the liquid here. Okay, so by looking at the color of the liquid, we can tell exactly what the plant is doing. There are mainly three main colors you need to memorize. Number one is purple. This, if the color is purple, this means the solution has become alkaline. It happens when carbon dioxide levels are very low. But why would they be low? Because the plant is photosynthesizing so fast that it is sucking up all the carbon dioxide. In this case, photosynthesis is dominant over respiration. So if in your exam you see a purple color which is alkaline, okay, then it means that photosynthesis process is greater than the respiration at that particular moment. Secondly is red or pink color. Okay, this is the neutral state. It shows the carbon dioxide is at normal atmospheric levels. This happens at a specific point called the compensation point, where the rate of photosynthesis exactly equals the rate of respiration. The third one is yellow or orange. Okay, you can see it here. This means the solution has become now acidic. It happens when carbon dioxide levels are high. So if you put a plant in the dark, it stops photosynthesizing but keeps respiring, that is pumping out carbon dioxide. So in this case, respiration is dominant over photosynthesis. Now let's put our knowledge into practice with a classic experiment. We're going to place identical leaves into 4 different tubes with hydrogen carbonate indicator and change the light conditions. In the first tube, you can see it here. In the first tube, we cover it with black paper. This one, okay, with black paper to create total darkness. Here, photosynthesis stops, but the leaf continues to respire. That is, carbon dioxide is released, the solution becomes acidic, and the indicator turns yellow. In the second tube, we use tissue paper. This one, okay, we use tissue paper. Tissue paper is used to create a dim light. This is a special state which we call as compensation point. Here the rate of photosynthesis is exactly equal to the rate of respiration. Since the plant is taking in carbon dioxide as fast as it is breathing it out, there is no net change. The indicator stays red. So in the third tube, we leave it uncovered. We leave it uncovered. That is, the plant is facing bright light. Now photosynthesis is much faster than respiration, and the plant is a carbon dioxide vacuum sucking the gas out of the liquid. The solution then becomes alkaline, as we studied in the previous slide. Okay, the solution becomes alkaline and the indicator turns purple. Okay, indicator turns purple. Finally, we have the control, usually a boiled leaf or an empty tube. Since the cells are dead or absent, no gas exchange happens. This proves that any color changes in the other tubes were actually caused by the living plant. This is the definition of compensation point. That is, the dim light tube represents the compensation point. This is the specific light intensity where the volume of carbon dioxide consumed by photosynthesis is it exactly matches the volume produced by respiration. A quick exam pro tip for you, that if you are asked why the indicator turns yellow, remember the chemistry. Carbon dioxide dissolves in water to form carbonic acid. More acid means a lower pH. Okay, if there is more acid, then pH will be lower, and a lower pH means a shift toward the yellow or orange color at the end of the scale. So always mention the pH change to secure extra marks. Now that we have understood how the indicator works, let's look at another classic setup which demonstrates the delicate balance between plants and animals. This experiment shows us exactly how carbon dioxide levels change when we have different organisms living together. As a quick refresher, yellow means high carbon dioxide, red is our starting neutral point, and purple means low carbon dioxide. So in this experiment, we are manipulating two main variables. First, we are testing light versus dark to see if photosynthesis can happen. Secondly, we are looking at the organisms present. So we have two very different players in our tubes. First, we have pondweed. Okay, this is the pondweed. Like all green plants, it is an expert at both photosynthesis and respiration. Secondly, we have a pond snail. You can look at the bottom. Okay, that is a pond snail. So unlike the plant, the snail is heterotroph. Why that is heterotroph? Because it cannot create its own food. Okay, it only performs respiration, meaning it is constantly releasing carbon dioxide into the water, whether it is light or there is darkness. So in the next slide, we will predict exactly what happens when we put these two variables, that is pond weed and pond snail, together under different lighting conditions. We shall answer a question that will the plant be able to clean up the carbon dioxide that the snail is producing? So let's find out in the next slide. Now let's look at what actually happens in our tubes. We will start with the bright light condition. Remember our rule that in light, plants photosynthesize faster than they respire. So this is a tug of war. The snail is adding carbon dioxide, okay, but the weed is so busy photosynthesizing that it absorbs its own carbon dioxide plus the snails, okay, means which the snail is producing. So the indicator turns purple. Now the next case is tube with weed only. Okay, this is the tube with weed only. There is no pond snail you can see here. This is very easy. Rapid carbon dioxide uptake for photosynthesis makes the indicator turns purple quickly. Then tube with snail only. This is the case. There is no plant in this tube. So if there is no plant, then it cannot, then the carbon dioxide cannot be cleaned up. The snail respires, adding carbon dioxide more and more, and the indicator turns yellow. Why it turns yellow? You will answer that question. I think you have understood that why it turns yellow. And yellow means acidic or alkaline. Okay, you can give your response in the comment section. So this is the control. There is no pond snail, there is no pondweed, okay, there is no life. If there is no life, it means there is no change. It will stay red. Now what will happen in darkness? This is where, this is the darkness, okay, condition. This is the darkness condition. This is where many students get tripped up. In the dark, photosynthesis stops. Keep in mind that when there is no light, there is no photosynthesis. So in the dark, photosynthesis stops completely, but everyone keeps breathing. Means respiration process is on its way. So if there is tube with weed and with snail, now there they are a team. Both are releasing carbon dioxide. Okay, if both are releasing carbon dioxide, then what will be the condition? Acidic. It means that will be that the indicator will turn yellow very fast. Then in next case is tube with weed only. Even though it is a plant, it is still a living thing. It respires 24/7. So as it is respiring, but without light, it is using carbon dioxide. So the gas builds up and turns the indicator yellow. It means under darkness, photosynthetic process stops. It means this pondweed is not consuming carbon dioxide. If not consuming, then definitely the concentration of carbon dioxide will get increased in this condition. So the indicator will turn yellow. Then in next case is tube with snail only. What will be the result? Definitely the result will be yellow. Why yellow? Because snail is respiring. Okay, that is giving out carbon dioxide, but that carbon dioxide is not being consumed due to darkness, number one, and definitely there is no plant which can consume that carbon dioxide. Okay, so the indicator will turn yellow. This experiment is the ultimate proof that plants only remove carbon dioxide when light is available. But respiration, that process never stops 24/7. Here we are wrapping up our study of gas exchange. Let's look at how plants and animals interact with the environment in different conditions. We use our hydrogen carbonate indicator as the judge. So under light conditions, okay, under light conditions, a plant is a carbon dioxide vacuum. Okay, because the rate of photosynthesis is greater than the rate of respiration, the carbon dioxide level in the tube decreases. So the indicator reflects this by turning purple. Purple, alkaline, photosynthesis is greater than respiration. Okay, remember this rule. However, if you swap that plant for a snail, the story changes. The snail only respires, adding carbon dioxide to the tube and turning the indicator yellow. Now what happens in the dark? This is where students often get trapped. Okay, but I hope you will not get trapped. Okay, as you have clearly understood the mechanism. So under dark conditions, the plant's solar panels are off. Those are not working. That means photosynthesis stops, but respiration continues. Just like the snail, the plant now becomes a carbon dioxide producer. Both the plant and the snail will increase the carbon dioxide levels, turning the indicator into yellow color. Now, what is the big takeaway here? Now let's look our theory into the lab, we want to answer a simple question. Does more light actually lead to faster photosynthesis? To find out, we use an aquatic plant like Elodia because we can physically see the oxygen it produces as bubbles in the water. In any good experiment, we have to define our variables. Our independent variables in this case are the things which we are changing. So independent variables are those which are in our control, okay, which are not dependent, which are in our control. For example, here is the light intensity is an independent variable. We control this variable, that is the light intensity, simply by moving our lamp. We are using this lamp as a light source, okay. So we can control this light intensity easily by moving our lamp closer to or further away from the plant. If you talk about the dependent variables are those variables which we are going to measure. And here what we are measuring? We are measuring the rate of photosynthesis. So rate of photosynthesis is a dependent variable. Since oxygen is a byproduct of the reaction, we can count the number of bubbles. You can see it in the bubbles producing bubbles which are being produced in the, okay, you can see that in this picture, okay. So we can count the number of bubbles released over a fixed time. For example, we can take a one scale, that is 1 minute. So more bubbles, if more bubbles are being produced, then the rate of photosynthesis is faster. But for this to be a fair test, we must keep our control variables constant. Because okay, what it will be the control variable? That is will be temperature. Why we are keeping that constant? Because temperature also affects the rate of photosynthesis. So we shall keep the temperature constant to eliminate the effect of temperature on this rate of photosynthesis. So temperature, how that will be controlled? Here is the solution, that a lamp can give off heat. So we often place a glass beaker of water between the lamp, okay. This is the glass beaker. It will absorb the heat intensity, okay. It will not stop the light, but it will eliminate the effect of temperature. It will act like a shield. So we add a bit of sodium hydrogen carbonate to the water here. Sodium hydrogen carbonate to the water. This will ensure the plant has a steady unlimited supply of carbon dioxide, so that it does not become a limiting factor. So in that case, we shall keep carbon dioxide as a constant factor. Here we shall use the same piece of pondweed here for every measurement to ensure the surface area remains the same. So by keeping these factors the same, we know that any change in bubbles is strictly due to the light. Why light? Because there are three factors: light, temperature, and carbon dioxide. We are keeping temperature and carbon dioxide as constant variable. We are controlling that. Okay, there will be no change in temperature, there will be no change in carbon dioxide, but we shall change the light intensity by moving light lamp away or further to the experimental plant. To get accurate results in the lab, we need a very specific setup. We use pondweed, as told you earlier. We use pondweed because it is one of the few plants that lets us photosynthesis in action through bubbles. We dissolve sodium hydrogen carbonate in the water to ensure the plant has plenty of carbon dioxide. We then place an inverted funnel. You can see an inverted funnel here. Okay, we place an inverted funnel over the weed to trap the oxygen bubbles and direct them into a collection tube. So to keep the test fair, we use a water bath as a heat shield to absorb the warmth of the lamp. This is the solution here, okay. This is the, you can see this is a water bath which is like a heat shield which can absorb the warmth from the lamp, and a ruler to measure our distance exactly. Now the process is very simple. Start far away, let the plant adjust and count the bubbles for 1 minute. Then move the lamp. I'm talking about this lamp. Move the lamp closer, okay. By moving the lamp closer, the bubble rate will be again counted, and we shall repeat this process at least for 3 to 4 times. Now look at the results. What will happen? If you plot distance against the bubble count, you get a curve. This is because light intensity does not drop in a straight line. You can see curve here, okay? uh, Light intensity is not being dropped in a straight line as you move away. It follows the inverse square law. So however, if you plot light intensity against the bubbles, you get a beautiful straight line. This is a straight line, okay? We are applying this formula here, 1 over distance square, okay? That will be versus equal to bubble count would show a straight line until another factor like temperature becomes the limiting. This proves that light is the limiting factor, at least until the line plateaus, because the plant is working as fast as its enzymes will allow. This is do not think that this is complicated. This is not complicated, okay? This is very clear. That I am again giving you just a brief note that this is a light lamp, okay? This is uh, the independent variable, that is light intensity, okay? If you are moving this light lamp away, or if you are moving light lamp towards further towards this pondweed, definitely it is affecting the rate of photosynthesis. And how we shall count the rate of photosynthesis? We shall count the bubbles being produced due to photosynthesis, okay? By counting these bubbles and by measuring this distance, we can easily calculate the rate of photosynthesis, okay? That wraps up our lecture on light intensity and photosynthesis. Next time, we will analyze the roles of carbon dioxide and temperature as limiting factors. Make sure to subscribe to this channel to keep mastering your biology curriculum with clear step-by-step lectures. Thank you for watching this video. Best of luck and goodbye.